So I think we should get started. Welcome. It's a great pleasure and honor to introduce today's speaker, Ed Lein. Ed is a senior investigator at the Allen Institute for Brain Science. He's also the director of Human Cell Types program at the Allen Institute. And Ed is also an affiliated professor at the University of Washington in Seattle. So Ed received his PhD from Purdue University, then did his PhD at Berkeley, UC Berkeley with Carol Schatz, and his postdoc with Fred Gage at Salk Institute, neuroscientist we're all very familiar with. And Ed is an undisputed leader international leader in creation of the largest scale anatomical cellular genomic atlas, which many of us we actually use daily. I don't think there's a day I don't click Allen Institute Atlas. So, any questions? You need to know some answers. He is the main leader of this program, including mouse Atlas, and which I used since I don't remember how many years now, and now more recently on very complex multi-model human and non-human primate atlas, all at the Allen Institute. And currently, he's leading large-scale brain initiative efforts, and use the cellular genomics as the main phenotype to understand the human brain and non-human primate and mouse. Now it's the past mostly, and he's really international effort. Hundreds of people are involved. Many. I'm always amazed. I'm a small part of this effort, but I'm always amazed. How do you write a grant with 30 PIs and all together and get funded and working really well? It's unbelievable to me, right? So it's just his leadership, his vision is unbelievable. This is I don't think there are many people in the world that can do this kind of complex and visionary things. So it's benefit the Allen Institute, but really to the community. It's a critical resource for the community, and his expertise encompass many areas: development neurobiology and anatomy, genomics, and also in neurological disorders. So currently, he is using. You will hear some of them, I hope. Using the cellular data and genomic data, try to develop understanding the brain disorders, especially Alzheimer's disease, but also develop therapeutics for these disorders, including cell type specific, deliveries and enhancers, and also gene therapy for treating brain disorders. So, welcome and thank you.
Thank you very much, Guoping. It's a little hard to live up to that introduction, I think, but it's really a pleasure to come and speak to you. I have many colleagues in the audience here and tell you about the things that we're working on now. I'm really going to tell you about three different things today. It's actually Guoping already sort of mentioned. The first is trying to leverage modern molecular tools to create a new map of the brain, a cell atlas of the brain. And I'll briefly describe what that means and how we're going about this. This is part of the NIH Brain Initiative efforts. The second is starting to actually put these tools to use. So it's one thing to create a catalog of cell types. It's another to actually use this as a reference foundation so that we can better understand the cellular molecular consequences or phenotypes of disease. And then the third is really that these atlases have produced the raw material to understand gene regulation, to be able to create tools for targeting particular cell types. And these then sort of open up the field for which species can be studied at very high resolution, or medicine, for precision medicine to target very specific cell populations. So on the first part of this, I really just want to acknowledge that a lot of this work on brain atlasing, cell atlasing, is done under the auspices of the NIH Brain Initiative Cell Census projects. These have been through a couple of different phases. The first phase was called BICCN, or the Brain Initiative Cell Census Network. This was really largely focused on the mouse brain and trying to create an atlas of the mouse brain with the new technologies at that time of single-cell transcriptomics, spatial transcriptomics, and brought together a lot of different groups together to try to really have this idea of building a next generation atlas of the mouse brain. At the same time, it was also funding work towards working in bigger, more complex brains, including human brain. And that was successful enough, actually, that led to the next phase, which is called the Brain Initiative Cell Atlas Network, or BICAN, which is largely focused on human. And so that's really going from pilot phase to the scale up to try to deal with the complexity of the human brain. And I'll be describing some of that. Really, so much in this field is driven by technology. And particularly in human brain, we've had kind of a lack of available technologies to study brain. Mostly we have access to postmortem brain, some rare access to surgically resected pieces of living brain. But it's been possible now to really harness a set of new technologies to be able to use those tissues well and use them also to improve the brain preparation that can be then used across the field at large. And I really think of this as it's provided a way to both classify cells, to characterize cells, and then to manipulate cells. And so the classification side is really single cell and spatial genomics. The characterize are using living tissues and connecting phenotypes to those transcriptomic types. And then the manipulate is understanding the regulatory biology, in particular going for enhancers that can drive cell type selective expression. So I'll be going through these over the course of the talk. The really key thing, especially for human, where we have so little ability to manipulate, is that we need a way to bound the complexity of the system, to classify cell types. And single cell genomics turned out to be an incredible way to do this. We can apply this to complex nervous system tissues. Cells have distinctive transcriptional profiles that lets you cluster those cells. Those cells are putative cell types that you can then use as a foundation to start to ask questions about the properties of these cells or develop tools to manipulate them. This has worked extremely well. I think that the epitome of this really still is the first phase of the NIH Brain Initiative, which had the entire consortium focus on one part of the brain, primary motor cortex, and really substantiate that a molecular classification could be generated that's robust, that corresponds with other phenotypes, so that this is a meaningful thing for neuroscience as a whole. And over time, this idea of a hierarchical, transcriptionally based classification has taken hold as the basis of a cell atlas for the brain at large. But we have a lot of important elements of this. We have the types of cells, we have the expression profiles, we have the epigenetic profiles, we have the local spatial tissue distribution of these that includes the proportions and their relative relationships. You can add then cellular anatomy, physiology, connectivity. It becomes really like the genome. You can start to layer on information on top of this. Furthermore, you can do it across species and align homologous cell types across species, so you really know that you're looking at something quite homologous when you're looking at a human versus a model organism. Fast forward only 2 years, the scalability of these techniques has moved from single brain region to whole brain. Really pretty remarkable. This really means, in the case of the mouse, the BICCN phase delivered on its promise of creating a whole mouse brain cell classification, as well as a whole mouse brain spatial map of those cell types. And this really substantiated what I think a lot of us have felt for a long time. The brain is way more complex than other organs. With these types of thresholds, we can define over 5,000 types of cells in a mouse brain, which is kind of astonishing. Most of these are neurons, by the way. At the same time, in the same time frame, we were able to produce a first draft of the human, where we looked at about 100 regions of the brain and identified about 3,000 types so far, so about 2/3 of the mouse as a draft. I think it over-delivered as a draft. But it really showed like these techniques are scalable. They can be used across species, they can be used in human, and we can start to get this kind of information pretty readily. So we're now in the next phase of this, the Brain Initiative Cell Atlas Network, and the aspiration of this has now gotten significantly higher. The goal now is to produce something akin to the genome, where this becomes a well-curated, annotated reference that is substantiated by lots of other information that the community can use as a reference in the way that you use the genome. You map genome data to the genome and transfer labels onto your data. You map single cell data onto this, and you transfer the labels onto your own data. and that this reference can then really serve the whole community, not just the single-cell genomics community, but try to get to the cellular circuit developmental and systems communities, neuroimaging, as I'll get to a little bit, disease consortia, the other big projects in the Brain Initiative, like the armamentarium that's trying to create genetic tools. This is the raw material for that. The Connects program that wants to get to connectivity among cell types. That's connectivity between these cells. And so, so really this is meant to be a brain reference, but then also to interface with other communities studying other organs, because using genetics as a basis for this, we're speaking the same language across organs, and so we can actually have a human body reference or a human cell atlas, if you will. Okay, so this next phase, I wanted to call out that this involves a series of projects. One of them that I lead, that Guoping was mentioning here, which I'll discuss, the human and non-human primate atlas, where we're really trying to create a brain-wide atlas across multiple species here, across human, macaque, and marmoset. There are also projects that go deeper on the epigenome and start to go into development as well. And I particularly want to mention that one of these projects is also in the Boston area on individual variation led by Steve McCarroll and Eva McCosco. So our project is, as Guoping mentioned, quite a complicated project. We really tried to take this challenge seriously. If we want to create a next generation map, this needs to be something which bridges different parts of the community and brings different data modalities into making this. So we brought together the team of experts in all these different domains that could collectively bring all the expertise that we would need to do this project. Of course, this includes heavy representation here at MIT and at MGH with Guoping, Nancy Canwisher, Bruce Fischel, and Eugenio Iglesias, for example. Many other in the imaging communities that we're trying to link to, experts in different non-human primate species, in patch-seq as ways of characterizing cells, in informatics, and in ontologies. The overall goal, and I'm just really going to go in broad strokes here just to sort of advertise that this is happening, is to go to a really comprehensive cell atlas across the whole brain. This atlas is meant to be done in parallel with identical methods across human, macaque, and marmoset, so that we can truly have a homology-based classification that we're building across the species, the main biomedical species in non-human primate. It's multimodal, so it will be multi-omics as well as spatial transcriptomics to get the cellular mapping. We want to link this to fMRI-based functional organization, which means that we actually have to map brains into MRI-based coordinate systems, which actually has a lot of challenges to do on the front end of brain preparation, but also to be directly linking functional organization with cellular molecular, which means that we do analysis on animals that have been functionally characterized so that we can make those links directly. So this is a very large project. We are trying to do it in sort of bite-sized pieces. Like I described, for a focus on primary motor cortex at the beginning of BICCN, we're focusing on the structures that make up the principal basal ganglia circuitry as the first step in BICAN. Lots of reasons for doing this: heavy disease relevance. We wanted to move away from the cortex that we've been focusing on so heavily to other subcortical areas that might be more interesting to study as well. And really bring this consortium approach so we can bring together all the different skills of this consortium to study everything from cellular diversity to species conservation to disease relevance. In practice, this means building classifications from single-cell genomics. In this case, multi-omics. We're profiling human and macaque, I'm not showing it here, but marmoset, and then linking this to the mouse. We can do integrative analysis across the species, which shows largely homologous cell populations, but there are places where there seem to be some species specializations. One of these was actually identified by phenocrenin here several years ago, the TAC3 positive interneuron population. There are some others as well. But actually, this is often the general gist of what we're seeing is that the cellular architecture down to a certain level of granularity is actually quite conserved, and you can quite easily map homologies across species. When you get down to the finest leaves and the more detailed things, then things start to get species specializations on top of that. Doing spatial mapping is a lot harder in bigger brains because our platforms are built for things mouse brain size. And nevertheless, we're trying to create a map of something as large as a human brain. So this creates obvious problems. The solution so far has been to take a patchwork quilt type of strategy. So if we want to map a structure, say the basal ganglia, we actually have to find the slabs that contain that. We have to break that structure into pieces that would fit in the commercial platforms, section each one of them in parallel, and then sample them. And so you can get an idea here that the marmoset fits on this very nicely, very convenient. Macaque, you start to have to break into multiple pieces. Human, you have to break into many pieces in order to actually cover that. But it works. You can put this together. You get a really nice spatial map of the cell populations, as I'm showing here in macaque. This works in human and marmoset as well. And so we get now the spatial organization by sampling across the full spectrum of the slabs that contain these structures. On top of that, macaque offers the opportunity to characterize properties of these cells. And so we also have a significant arm with a technique called patch-seq, which is the culmination of patch clamp electrophysiology on living brain slices with sequencing of the contents of the cell. So you patch onto a cell, you record from it, you fill it with the dye. At the end, you extract the nucleus out and you sequence it. And that allows you to pin the phenotypes onto that molecular taxonomy, to annotate the taxonomy, if you will. And this is way more labor intensive than the omics. It's one cell at a time. But you don't have to sample super densely in order to get representation of the different cell types. And so systematically, we're being able to capture the anatomy and the physiology of these cell types. In some cases, there are obvious species differences. For example, the cholinergic neurons, interneurons in the striatum, are much, much larger in monkeys than they are in mouse. And you can see that by eye here. You can quantify it. And also properties like capacitance go up with the changes in size as well. So we both get to see what's conserved and also where there are some significant species differences. OK, so now to really make this a community reference, this needs to turn into a more formal ontology where we have the community helping to vet and annotate the platform. We're trying to develop robust taxonomies with standards and ontologies and share these data out to the world. This is actually really bolstered by working with Chloe Villani here from her work on the Human Cell Atlas to use some of these tools like the cell annotation platform, or CAP, that we're kind of beginning now. And imagine this is just the first stage of this. This is going to march across the brain to try to cover all the different brain regions and get as much annotation as possible and get those communities to buy into a classification. Because the goal at the end of this is that we actually have created something that can be a community reference. And in many ways, I feel that we're following in the steps of the genome, where as the genome became a useful reference, it became standardized. You had versions of the genome. You had formal nomenclatures associated with this. You had tools to visualize the data and mine the data. You had tools to map against it, like BLAST. And we have now tools like this. We have one called Map My Cells that maps your data in and transfer the labels onto your own data. So you don't need to de novo create a classification. Now, every time you do a single cell study, you build on this knowledge base and you actually can contribute to it as well. So that's what I just wanted to give you a flavor of what this looks like. The field is still in a lot of flux, I would say. Like literally every paper redefines the cellular universe in neuroscience. But hopefully that's going to start to change as this becomes more formalized, and maybe even journals will start to enforce the use of it. So we're not making this just to stamp collect. We're making this so that this becomes a useful reference for understanding biology and understanding disease. And so I want to spend the next part of the talk talking about how we can actually use this well-curated, annotated reference to better understand Alzheimer's disease. This is part of another consortium project that I run this one all in Seattle. That's a collaboration between the Allen Institute, the University of Washington, and in particular their Alzheimer's Disease Research Center, and Kaiser Permanente, which runs another longitudinal Alzheimer's or aging and dementia study. And really the aspiration here was to really directly use this reference to be able to profile what happens over the course of Alzheimer's disease and interpret it at a very fine level of cellular resolution to understand, which kinds of cells are vulnerable, which molecular pathways are perturbed in which cell types. Can we define Alzheimer's progression? And can we identify new cellular or molecular targets other than amyloid and tau as things that we might think of as treatment venues? So the tools that I've just described, of course, sort of more colloquially, we can think of as effectively defining what's affected, where it's affected, and when it's affected over the course of disease. So to do this, well, actually, one other point I just want to make. My own aspiration here is to bring this resolution that we have in cellular and circuit neuroscience to our understanding of human disease, which we really don't have right now. But we have the tools to do it. Once you can map to this kind of reference and you have the information about these cell populations, you can now interpret this at this very fine level. So now to do this was not trivial because it really requires high quality brain specimens. And we worked very closely with a colleague at the University of Washington named Dirk Keen, down here in the corner, who adopted some methods that we had developed to better prepare post-mortem brain specimens and implement this at a brain bank level. And so working with the Alzheimer's cohorts that I'll describe in a moment, we're able to prepare very precise thin slabs that allow precise anatomy, well-frozen material that can be used for either single-cell methods or spatial methods that require really intact histological tissues. And this has really made a huge difference. And actually, a little unwritten thing, these paper packages that have come out on the human tissues coming from the Brain Initiative all came from tissues that were from this one brain bank. So it's extremely effective for all these tissues. Not saying that there isn't good tissue elsewhere, but these methods can be improved quite dramatically. So these are cohort studies now we have to have in order to understand the progression of disease in the face of quite a bit of heterogeneity. And so in this particular case, we built cohorts from the combination of two Seattle-based cohort studies. One is the Alzheimer's Disease Research Center led by Tom Grabowski at the University of Washington. Like all the other ADRCs, typically people come in when they're symptomatic to an ADRC, and then they're tracked over the course of their life. You have clinical history. They donate their brain at the end. In contrast, a project called the Adult Changes in Thought, or ACT, is run by Kaiser Permanente. And this is a population-based study where you enroll at 65 or older with no dementia, and then you agree to be cognitively tested and tracked over the rest of your life. This turns out to be not really an aging study, it's an age study, because the average age of people that meet these criteria was actually about 89. So hopefully we all make it to 65 without dementia, because maybe we'll live for a long time. But at any rate, these two gave a really nice cohort. A really key concept that we approach here is that we don't want this to be a case control study. Disease is heterogeneous and it's progressive. And we should treat that progression as a continuous variable and try to really capture this and capture a donor cohort that spans the spectrum of pathology. So we did this with a cohort of 84 individuals. Some of these have just 80 pathology, and some of them, well, actually, what's more common than not is having comorbidities along with the 80 pathology as well. This really tracks across more conventional staging metrics like Braak staging, Thal for amyloid, CIRAD. And so it does seem to have captured the spectrum of disease from no pathology to late stage Alzheimer's. We wanted to track this both as a course of severity, by which I mean increasing pathology, but also across the brain. This is, tau moves from hippocampus, entorhinal cortex into the temporal lobe and subsequently into the rest of the brain. We want to see what's happening over the course of that progression. So that really meant focusing on a few brain regions earlier that then move into this whole progressive set of structures that typically we see tau moving across from the hippocampus up along the temporal lobe and then moving out across the cortex. So it becomes a rather big project across a cohort with many different brain regions. With this cohort in mind, now we can throw our whole toolkit at this. And so you won't be surprised, this includes the single-cell genomics, which is 10x RNA-seq, which is turned now into multiome, as well as the spatial methods. And this took some technical work to get these to work in donors that were up to 100 years old with high pathology. But actually, these techniques can all work on that tissue very well. We wanted to combine this with more traditional neuropathology, but turning this into a real quantitative metric instead of a staged qualitative metric. So I'll describe this as well, but this is really a key thing that in the same donors, in the adjacent sections, we wanted to capture the pathology as well as the molecular and cellular spatial elements. So this actually works very well. We developed a series of markers for proteinopathies, so tau, amyloid, TDP43, synuclein, as well as a set of cellular markers, and developed machine learning approaches to be able to quantify signal from these histological images, and does seem to do a good job of capturing the local burden of pathology. So within a particular Brock stage, for example, there can be a wide range of pathology. Brock doesn't capture that whatsoever. It only captures the regional distribution. This captures the local burden, which should correlate with whatever it is that we're measuring. We then build pseudo-progressions based on this sort of cumulative neuropathology to be able to model disease progression as an exponential function rising in pathology and order the donors along this trajectory. We can build this on the quantitative neuropathology metrics, but it correlates really strongly with other things like cognitive status, of staging criteria for ADNC, for example, or Thal or Brock does correlate very strongly with those. So then when you really look at this, it really seems to define two phases along the pseudo-progression. One that's really quite linear accumulation of pathology, and from a clinical perspective, typically preclinical, and a later stage where things start to become an exponential rise and become much more associated with cognitive decline as well. The next challenge was to map this data against the Brain Initiative's reference taxonomy. And so the idea was we're mapping this whole cohort, but let's map it all against this reference and transfer labels. But actually it was a bit more complicated than that because now we have a much bigger data set than we had in the reference. So that actually embellished the reference. So we ended up getting way better coverage, for example, of non-neuronal cells than we had before. We get certain disease states. And so the taxonomy actually gets bigger as we get more data. And we end up with a classification that included 139 types of cells that we can now start to measure. So now things start to get a little bit interesting with all this setup. So now we can ask what cells are actually lost as a function of disease. And so this is a proportional analysis as a function of some metric across all 139 populations. So I'm going to show this a few times, so I'll just make sure that I kind of guide you a little bit. On the left-hand side, these are the inhibitory interneurons, the GABAergic interneurons of the cortex, then the excitatory neurons in this block in the greens, and then the non-neuronal cells over on the right. We would only expect to see decreases in neurons as a function of disease, but we can get increases in cell states associated with non-neuronal cells. And so, you can kind of see that there's if you look as a function of some more traditional metrics, if we look at cognitive status or we look at a qualitative ADNC criteria, you can see that there's a lot of cells affected, but there also are a lot of cells that are not affected. But if we look as a function of the local quantitative neuropathology, the effect sizes get way bigger and rather more specific. So, for example, we can see that these are the the cortical excitatory neurons in the upper layers of the cortex that are affected. Many of the types of interneurons, but by no means all of the interneurons are affected. Many of the excitatory neurons are really not affected in the deeper layers, for example. On the non-neuronal side, there are robust effects of disease-associated states of astrocytes and microglia. These have been both described very well before, so we clearly see that. But we actually also see some things like loss of particular types of oligodendrocytes as well. And just a teaser for what I'm going to show you more about, if we look in another cortical area, we see roughly the same pattern. So it's starting to look like a pattern of cell vulnerability that's somewhat consistent across the cortex. Okay, so we had actually published that part. And now we've been working on the harder problem of looking across many more cortical areas, and actually not just cortical. So as I mentioned before, we've profiled a number of other areas that are part of this progression of tau aggregation and sampled across all of these. If we look at the pathology, this is probably not a surprise to aficionados in the audience, but amyloid and tau behave very differently. So tau is progressive. You initially get tau deposition in the hippocampus, entorhinal cortex, and then it moves through the temporal lobe, and eventually, at the very late stage, gets into visual cortex. Amyloid, on the other hand, is much more sort of you have it or you don't have it. And so we have a number of donors at the low end that really don't have any, and then it sort of gets hit or miss in different areas, and then you have it sort of across the brain. And you can kind of see this in a cross-correlogram as well. All the regions are highly correlated for the amyloid and neighboring regions for the tau. This is actually pretty interesting when you start to look at this regionality and how it relates to dementia status. You can have significant tau, but if you don't have amyloid there, typically unaffected cognitively. Once tau gets out of the temporal lobe, you essentially always are getting this. So, and you always have amyloid in that case as well. So really you need the combination of these two. And I don't have time to describe this at the moment, but it's really consistent with an idea that the tau sort of moves up through the temporal lobe, but it stays there unless amyloid is there, and amyloid becomes sort of permissive for tau deposition to then escape the temporal lobe, if you will. And that's really not a good thing. Okay, so building taxonomies is a lot harder when you have many regions. We were trying to build a cross-regional taxonomy that we could then ask questions about cellular vulnerability. This is because different parts of the cortex have quite different signatures, and hippocampus even more so. We've published on this before. This super complicated plot is really showing that most cortical areas, this is looking about eight cortical areas, have very similar GABAergic interneuron makeup. They can have really quite different excitatory neuron makeup, especially in the input layers of the cortex. Primary visual cortex is the outlier in this. It's really different. Of course, we know this structurally, but it turns out cellularly it has really different cell populations as well. So really trying to, and the hippocampus and entorhinal cortex have actually cells that would cluster differently. They're not really similar to the neocortex. So we put this together. We've built now a cross-regional taxonomy that integrates very well across the cell types that are common across the cortex. It has regional specializations, for example, in the entorhinal cortex or the parts of the hippocampus. So it's really covering all of these different structures, but in a way that we can be largely comparing like populations across the different regions. And hit you with hopefully the most complicated picture of the thing. This is sort of the nature of the business these days, is that we have huge amounts of data to try to digest and visualize. What I want to first of all highlight that this is looking across a subset of these areas that should be in the progression of tau deposition. So hippocampus, entorhinal cortex, the middle temporal gyrus that we started with, superior temporal gyrus moving into frontal cortex, and finally V1 typically isn't really thought to be involved until very, very late stages. And first of all, it's incredibly conserved in terms of cellular vulnerabilities across the different cortical areas, like almost shockingly conserved of which cell populations are affected. So it really seems to be somewhat of a canonical cortical set of vulnerabilities to AD pathology that affects specific cell populations. And those largely are the cell populations I've called out before of the cortical upper layer neurons and a variety of different types of GABAergic interneurons. The same is true on the non-neuronal side as well. Of course, there are regional specializations. It's specific types of CA1 and other hippocampal excitatory neurons that are lost, and specific types of upper layer entorhinal cortex neurons that are lost. And so we can really pin down which cell types are being affected. Now, there are surprises in here too. It turns out that there are similar vulnerabilities in primary visual cortex, even though this is typically thought to be a somewhat unaffected area. And actually, some of these should have phenotypic differences. So there's a loss of the input layer receiving visual input from the thalamus in primary visual cortex at the end. So maybe by that time, we're not so sensitive to giving tests to people for their vision. But this would certainly suggest that there are some visual deficits that would arise at late stage. OK, so now it's starting to try to dissociate which things are related to amyloid and which things may be related more to tau. One thing for sure, these disease-associated microglial states are only seen in any area once you have amyloid. So if you have no amyloid, you have no disease-associated microglia. So they seem to go together. But now if we ask the question about, are these cell vulnerabilities that we're seeing more related to tau or more related to amyloid? It's a super clear answer. They're way more related to tau. Tau seems to be the really toxic thing here. Now you need amyloid for it to get out into the rest of the cortex, but it seems to be tau that is probably the really destructive part of this pathway. OK, the last part of this story, but I think actually one of the most interesting parts, is when we start to look at the timing of events. I've only showed you what's affected. I haven't showed you when it's affected. And so now, because we have these pseudoprogressions, we can ask, when along the pseudoprogression are these cell losses happening or cell changes happening? And so if we look in the temporal cortex again, you can see that the astrocytes and the microglial states associated with the disease really start to go up early, and they keep going up over the course of disease. But when we look at the neurons, we get a somewhat different picture. Most of the neuron loss, which is well documented in the literature, of, for example, the cortical excitatory cells, happens very late in this progression. In contrast, one of the types of GABAergic interneurons, the somatostatin-positive interneurons, is lost very early in disease. And I made a point of sort of showing this early and late epoch earlier because this loss is happening well before there's much pathology in the brain. So it seems to be a very early hallmark of disease. It seems to be something that's seen across different cortical areas as well. We see a very similar thing if we look in the frontal cortex. Now we're looking at other parts of the brain as well to look at the relative timing of events. So, this is kind of an important result that we're starting to see certain types of neurons that actually are affected way before the things that have been described before. We have now investigated more about what those cells are. Throwing merfish at this, we can then ask where in the cortex are these cells found. And it turns out that the vulnerable cell populations, the ones being lost, are really all up in the upper layers of the cortex. So it's not, the somatostatin neurons are distributed across the whole cortex, but the affected ones are in the upper layers. And that puts them interdigitated with the cells that are lost subsequently. So most of the action is in the upper layers of the cortex. The other types of inhibitory neurons that are lost later are also in the upper layers. The long-range cortical neurons that are lost are in the upper layers predominantly. So this really could be something of a chain of events where some of the neurons are lost that are making connections to others, and subsequently you get others, at least as a hypothesis to test. And then finally, we know what these cells look like now as well because of the annotation effort on the cell atlas. So these somatostatin neurons are well known to be really phenotypically diverse. These are actually human morphologies here that you're looking at from surgically derived tissues. And they include a very specialized subset that's been known for over 100 years called the double bouquet cells. This is a primate-specific morphological specialization. So, we're really getting down to the circuit level here, but also in a way that you could not have found in a mouse. So you couldn't have gone straight to the model. You need to find the human phenotype and then see if the models actually recapitulate that thing. And that's a point I really want to make, that I think that we need to be thinking that way more. If we want to tackle a disease, you first have to understand what's actually happening in the human and then go try to model that thing, which is actually not how things mostly go these days. This really could change thinking about phenotypes of disease. These somatostatin neurons are well known in the circuit field for their inhibitory connection onto both excitatory and inhibitory neurons, so they're a really key place to be controlling neuronal excitation. There's well-known hyperexcitability, even seizures in Alzheimer's, that this could be related to if you remove inhibition. They could actually disrupt trophic support to other neurons, and that could be part of why the other neurons are lost later in disease as well. So lots of new ideas that can come from this. And one that I particularly like is the cell now can become a target for thinking about therapeutics if we can target the vulnerable or affected cell populations. So I just say that I've cherry-picked just a few things from this study. When you look at 139 cell populations across 10 different cortical areas with omics, it's an enormous data set. Really get a holistic picture from these data sets about all the cell populations that are affected and their sequence of events. And different molecular things are happening in all different kinds of studies. In fact, there's often in the field, we talk about which is the more important mechanism. They're all important and they're all there. They're just there at different times in different cell populations. And this is all available publicly as a data resource if this is of interest to any of you in the audience. Okay, I'm gonna end the talk on a slightly different topic, one I brought up at the beginning, which is that these cell atlases, and particularly the epigenome data, have provided a really clear and straightforward path to identifying regulatory sequences that can drive selective expression in a viral vector system. And so effectively we have attack-seq data, which illuminates regions of open chromatin in particular cells. Often these are very cell type specific. And when we first started seeing this, we realized that these really have the hallmarks of a distal enhancer when these short cell type selective peaks. So we set up a screening platform to stick these into adeno-associated viruses. AAVs can be put into a mouse as a screen or other tissues and with a reporter, and you can then look for cell type specificity driven by that enhancer element. We can screen for these in mouse. We can take the hits that work and take them into non-human primate. Typically, this is macaque in our case. And they can be quite dramatically improved as well through things like concatenation to improve their robustness or their specificity. Just to briefly lead you into this, these combine really well with advances in capsids, like the PHB-EB capsid that crosses the blood-brain barrier in mice. So you can do a retroorbital vein injection and get brain-wide transduction, and then the specificity is driven by the promoter or enhancer. And so here is a brain-wide synapsin promoter to give all neurons, DLX to restrict it to forebrain inhibitory neurons, get way more specific with things like just CA2 excitatory neurons across the brain. And we've now really extended this to be able to systematically deconstruct circuits in different parts of the brain and develop tools to target each of the main cell populations. I'll come back to the striatum since I used that as a point earlier. We now have tools to target all of the medium spiny neurons, the main neuronal population, just the D1 or direct pathway, just the D2 indirect pathway, the ventral striatum populations, the different interneuron GABAergic populations or cholinergic populations that make up the system as well. So this can now be done anywhere. We can do this in any part of the brain to just march across the cell populations and develop tools for their selective targeting. These become immediately really useful research tools, and often you can now combine them as well. So here's an experiment matching 3 different interneuron populations in a single experiment. This could be with one to excite, one to inhibit, all sorts of molecular genetic tricks that can be done in any species. It could be done in a macaque, it could be done in a marmoset. So really opening up the tools for being able to do manipulative cell type selective biology across species. These typically, actually at that level of resolution, are conserved across species. And so we take these from a mouse screen into a macaque. In this case, I'm showing stereotaxic injections where a direct pathway medium spiny neuron enhancer is injected into the striatum of a macaque. And so you can see both striatal expression as well as substantia nigra, where the projections go. So, so very often we can quickly translate to something which works in non-human primates, which means it probably works in human as well. Just a little highlight reel, we can start to target all kinds of cell populations that have clinical significance right away. So the neuromodulatory pathways like dopaminergic, serotonergic, noradrenergic, for example. We can tackle the motor neurons in the spinal cord and selectively be able to manipulate those. We can tackle the double bouquet cells that I mentioned for Alzheimer's disease. So this was actually a bit of serendipity that we didn't expect. We weren't trying to target double bouquets, which don't exist in mouse, but it made it through a primary screen, and it turned out to do this. So never cast shade on serendipity. We can get some very useful tools from that. So you could imagine that targeting these to try to prevent their degeneration could actually be a potential clinical application of these in an AD case. Okay, I want to just end expanding on that topic. These really become tools that could be used for precision therapies, where there's a cell type vulnerability or a cell type locus of the phenotypes of disease. And so we've been trying to apply this to several different diseases in the genetic epilepsy space, where it's known that there is a GABAergic interneuron locus of a disruption of the EI balance, excitatory-inhibitory balance, that leads to seizures and other phenotypes of disease. One of these is Dravet syndrome. This is a childhood epilepsy caused by a haploinsufficiency of the SCN1A gene. This is causal in most cases of Dravet, and it's really not controlled well by medication at all. So there are no FDA-approved gene-restoring therapies to this, and it's quite well known that the inhibitory neurons are really dysfunctional in this particular disease. So we have terrific tools for targeting the inhibitory neurons selectively. These are the GABAergic neurons. This was actually an embellishment of a tool that has been around for a long time for a DLX enhancer. John Rubinstein made it initially, then Gord Fischel worked on it, and then we improved it even further by making a triple concatemer that really improved the expression levels. And it works everywhere we throw it. It works in mouse, it works in monkey, it works applied to a human neurosurgical slice that just came out of a human being and still selectively labels inhibitory cells. In this particular case, there was an extra challenge to this. The SCN1A gene is too big for an AAV capsid. This is a very common problem with these kinds of things. But it was big. It was small enough to fit with a 2-part system. And so we developed a split intine system to be able to reconstitute functional SCN1A gene in cells that are doubly infected. And we can really clearly demonstrate that we're able to get a full length and demonstrate that it's a functional protein. And when we do this, we get really pretty striking rescue of some of the phenotypes of disease. So for example, the SCN1A knockout mice have a strong infant mortality syndrome that is completely rescued by restoration of this SCN1A gene. They have a very strong heat-induced seizure phenotype that's almost entirely rescued by this as well, but not if you put it in all neurons. So this works if you put it in just the inhibitory neurons. If you put it in all neurons with a synapsin type of promoter, it's actually worse on the mortality. You can get a seizure rescue eventually with a very high dose, but you're not able to rescue the mortality part. So you apparently really need the cell selectivity in order to get this without any off-target effects. We've now also achieved this in a second genetic epilepsy, SLC6A1. So this is a reuptake loss of function. And interestingly, the SCN1A gene is expressed normally both by inhibitory cells and also in astrocytes. And so we've tried now replacing a functional copy in either the inhibitory neurons or the astrocytes. I'm showing this in a little bit vague fashion because some of this is still a bit not open for disclosure yet. But we're able to completely rescue the mortality phenotype and seriously decrease the seizure phenotypes as well with rescuing actually either of these cell populations. It can work either in the inhibitory cells or the astrocytes. So both of them are involved in removing GABA, and it seems to be sufficient in either case to do that. So these are meant also just to be illustrative cases of what we can do now with this. If we can identify the cellular locus of disease, we can target it now. And this is probably the lowest possible bar of a gene replacement. But you can now imagine all kinds of acute manipulations. You could do DREADS, you could do channel rhodopsin, you could do all sorts of manipulations with this now that you can target the right circuit elements. And so I'll just leave with that. I think hopefully I've convinced you, illustrated to you what it means to make these cell atlases, but also maybe some of the places that they can be put to use right away. And we've been really involved in the simultaneous creation and trying to use these atlases, but they're ready for any of you to use as well now, either to apply to disease to better understand the disease, or to think about targeting the relevant cell populations that might be affected in disease. So with that, I'll end, and thank you for your attention. These are big, just a moment please. These are big team science projects. I really want to call out that there are many, many people involved. I'm showing here the teams at the Allen Institute. We often have cross-matrix projects that involve many of these people, as well as the two projects I've already shown, sort of headshots for the CAD and HMBA, and very, very generous support from our various funding, and also really from the donors and the surgeons who've been willing to contribute tissue to these studies as well. Thank you.
That's a great talk. So for the cell vulnerability in the AD, I have two questions. The first, do we know why part of the cells, especially the integrons, are more vulnerable to the disease? The second is, do you think there's so far there's a causal evidence showing the causal relationship that their SSD loss on the early onset has been lead to the PV1, the layer 2 to 3 loss later?
No, these are great questions that I think, in some sense this is a new finding, in some sense it's an older finding. Others have suggested that there's somatostatin decreases with other types of techniques, but I think it's totally unclear why these cells are particularly vulnerable, what the insult itself is actually, or what the downstream consequences of this would be. But you can imagine now that you could try to go into macaque and remove those cells. Would that lead to loss of the other cells? You could test that kind of idea. It's also possible that this is not a primary effect. And what I mean by that is that another long-known fact is that some of the neuromodulatory inputs are lost very early in Alzheimer's, like the noradrenergic system and the cholinergics, like the FDA-approved treatments for cholinergics. This could actually be secondary to that. which could explain why you get a really broad loss across the cortex of those cells. But the fact of the matter is that this hasn't been demonstrated in a mouse, and so it hasn't been available to study. But I think this is what I mean by human first kind of phenotype. Like once we have this, we can start to ask those questions or figure out an experimental path to ask those questions. Yeah.
And absolutely beautiful work. So it's nice to see such a crazy project coming together so beautifully. So I love every part of the talk. I'm just going to ask a quick question on the last part, which is, it seems very, very nice to be able to do that in such a selective way. And my question is, a lot of the textbooks basically talk about these enhancer domains in these large regions. And it seems that you're taking these relatively small segments and they kind of work. So I'm wondering, is the large-scale effect mostly a repressive effect? And therefore the enhancers would be sufficient to sort of activate things, even if they're adoptively delivered? And I have several questions about sort of the size, basically how big they have to be, about whether they integrate or not, and how that affects expression, and whether you can package them in specific ways that will sort of perhaps integrate them in specific loci and so forth. So if you have some questions, some comments on what have you tried and what has and has not worked.
Yeah, that was a lot of questions in one. Okay, let me start from the basics. So first of all, they do tend to be quite short. So we typically start with about a 500 base pair piece, and that's the starting point for doing a screen. If we want to improve it, we bash it, and we typically bash it into 3. And the activity is typically contained in one of those. And so actually, it's generally within, say, 150 to 200 nucleotides that all the action is. So these are very small elements that really can only have a handful of binding sites in them, I would think. We're now actually starting to use generative models on sequence models on this, and can predict now which parts of these are likely to be the important ones and are surprisingly effective at that. Ideally, we'll get to understanding the motif structure and why this is happening. I'd love to see a path to synthetic enhancers here, where we're just completely de novo creating these to spec of whatever we want. Now, they can often be way more specific than a gene's expression is, and I think that's because a gene is under the control of many of these things. And so it could be multiple activating things, it could be some are activating and some are repressing. We don't really have access to that information because we're only taking this piece, but it's shockingly effective. Like, we don't have to multiplex screen these things. We do them one at a time, and it's a high enough hit rate that's effective. So that seems to be sufficient in of itself. Now, for me, it's all about the sequence, because we can get things. We explored for a while. Like, should we look around marker genes? Should we look anywhere in the genome? Should we look whatever? We tried them all. They're good examples of things that work in all those cases. We tried to use homology. Like, do they have to be open? Do they have to be conserved? They have to be open across species. And the answer is no, not really. If it's functional in a mouse and not open in a human, or vice versa, it can still work in a mouse or a human. So it's being open is not the same as whether it can be functional. I don't think these are integrative. I think they do concatenarize in a cell, but AAVs gutted of everything are not the same as the native AAVs, which does have a site-specific integration. So I think that they're basically acting as episomes.
And how big of a payload can you deliver?
Well, it's still 4.7 kb with, there's some wiggle room on that. You can sometimes do oversized, but that's pretty much it. So the good thing is that they're short. Now, often what we do is, the native one might be specific but not very robust, and so then you can concatenarize it, and that sometimes gives you a really nonlinear increase. And so now you're back to 500, but still that gives you most of the capsid for your therapeutic payload or manipulative payload. Yep. Please.
My question is with reference to your consensus cellularness of the basal ganglia. Has addition of another modality, specifically the genomics data, refined or added more granularity to the cell type we would have otherwise identified with just, say, single nucleotides?
Yeah, it's a great question. So we're actually right in the midst of doing that analysis right now. So we have ATAC. ATAC is pretty sparse, and so for the most part, we don't usually feel that we get very much extra information from that. We get very useful information from it. But Joe Ecker and Bing Ren have one of the projects in this, and so they're going way, way deeper on methylation and other epigenetic marks. And for the most part, it still is lower resolution, and they just are mapping to the transcriptomic one, but not exclusively. They're finding some things which you only see in the epigenome. And so now we're figuring out how to add that to the reference as well. So it's a work in progress, but I think there will be cases where the epigenome has more information. I'm sure of it. Yeah. And maybe if that data can be made less sparse in the future, there will actually be a lot more information there. But it's much sparser than the transcriptome. Yep.
I'm curious about the AD part. And it seems, I don't know whether it's true, like women seems to have a later onset of AD. Do you find any gender difference in your data set? Like maybe some genes X-related or Y-related?
Yeah, we didn't see huge sex differences actually, but I think we wouldn't be able to ask that question because this was, as I mentioned, it was an aged cohort. I think if it was an aging cohort, we might have seen that more obviously. But there was very little information in the aging because it was mostly 85 to 100. So it seems from everything else as you're citing there, I mean, we should see some differences, but they're not huge. At least in that cohort, they're not huge.
Quick questions. Is your brain map, included cerebellum? In Alzheimer, there's the cerebellum, particularly the posterior lobe, the vermis, and also some of the pathology may share there, and also the sleep disturbance, and some of the cognitive circuit connected between the cerebellum and the cerebrum, also affected.
Yeah, so we haven't gone there yet. I mean, so this is one of the problems with these survey projects, doing it across a cohort, that you'd like to be able to look at many regions. I think this survey told us a lot. It was very cortex-centric. Where we're hoping to go in the next phase is a lot more subcortical things. And so, and as new areas come out, the sleep nuclei are kind of a new target there to think about. I want to hit all the neuromodulatory systems as well, for example. So I think it's really important, and I'm sure there are effects deeper in the brain. We just haven't looked there yet.
Thank you.
I had a quick question, which is like clinically relevant, whether you have enough Alzheimer's cases to see whether there are subtypes with your data that clinicians might not have recognized.
Yeah, we're really not powered for that yet. We tried to take the tack that there was one dominant trajectory, and that worked. But I think we know there's way more heterogeneity than that, and but once in this size of cohort, we just don't have enough to really tease those things apart. So in the future, we've got to get. We've been talking about this. We've got to get to way larger cohorts so we can start to stratify different mechanisms, but not so far.
One last question. Just building on the prior question about incorporating genetic information into your cell type classifications, what is your guys' thinking about incorporating other domains of cell identity, like for example, structural morphological features, projection patterns, electrophysiological firing patterns? I imagine those are harder to kind of scale to the same extent as the single cell, like genetic profiling, and then to map it back onto genetic profiles. But I guess I'm just curious, where are you guys in terms of taking those features of cell identity and mapping them onto genetic classification?
Yeah. So for the most part, I mean, my philosophy at this point is that the molecular approach to cell classification is descriptive and it's predictive. The genes predict things that should happen. They allow you to do a mapping across organs. They allow you to do mappings across species. They allow you to understand why they might be there and what their properties are. So the other properties are they're properties of cells that were derived from the genes that were used by those cells at some point. Now that may be not totally obvious in the current state of a cell, but there's so much value in being able to do that so far we've been taking the strategy of classifying on that basis, then collect other phenotypic information and see how well it correlates. The field at large has been trying to use things like physiology to classify cells or morphology, and it's been a giant mess that goes nowhere. Like, this is really a unifying way of thinking about these things. Although there may be mismatches sometimes, there may be heterogeneity in a particular phenotype within a molecular class. So that happens. We have to accept that. But there's only one unifying framework that we have right now that can work for everything, and that's the genetic framework. So in that sense, in my opinion, it has primacy, in part because of its explanatory power in the sense of a genome, in part because of its extreme pragmatic utility as well. But we'll learn more as we know more. As you have this framework, you can start to characterize cells, and you'll see where there are good matches and where there are good mismatches.
Just make one comment. Ed said about the subtype. Ed said something really important, which is to think of disease as longitudinal. And one of the reasons that everything is in some ways underpowered is a lot of treatment relevant subtyping and pathology only appears over time. Like, who gets TDP43 after X number of years of amyloid and tau. And you get your brain once, right? So you need a large enough cohort to understand these things longitudinally. We have the same problem in psychiatry. There's so much diagnostic instability. We recruit people into a imaging study for bipolar disorder, and then 5 years later, the same person has a diagnosis of schizophrenia. So I think the importance of these longitudinal studies, and I think you said it perfectly, it's really important.
Great. So we can continue discussion during the reception. Let's thank Ed again for a great seminar.